Hello everyone. Today we are discussing about enzymes. What are enzymes? Enzymes are specialized proteins that act as biological catalysts in living organisms. They speed up chemical reactions within cells, without being consumed in the process. Enzymes are involved in almost all biochemical reactions in living organisms, from digestion to DNA replication and synthesis of new molecules. They are highly specific in their function, meaning that each enzyme is designed to catalyze a specific chemical reaction, and will only act on certain substrates or molecules. Enzymes play a critical role in maintaining metabolic homeostasis, regulating cellular functions, and sustaining life. Definition of enzymes. Enzymes are biological molecules, typically proteins, that act as catalysts to accelerate chemical reactions in living organisms. They are highly specific in their function, catalyzing only one or a few reactions, and can increase the rate of chemical reactions by factors of up to several million. Enzymes are essential for life and play critical roles in many biological processes, including digestion, metabolism, and DNA replication. Do the enzymes are protein? Yes, all enzymes are proteins or protein-like molecules. Enzymes are composed of long chains of amino acids that are folded into a three-dimensional shape. The specific arrangement of amino acids in the enzyme determines its unique structure and function. Some enzymes may also require non-protein components, such as cofactors or coenzymes, to perform their catalytic activity. However, the protein portion of the enzyme is always necessary for its function. In summary, while there are many types of catalysts, enzymes are a specific type of catalyst composed of proteins or protein-like molecules. Characteristics of enzymes. Enzymes have several characteristics that distinguish them from other types of catalysts. Specificity. Each enzyme is highly specific in its function, meaning that it catalyzes only one or a few closely related chemical reactions. This specificity arises from the unique three-dimensional structure of the enzyme and its active site, which is the region where the substrate molecule binds and the reaction takes place. Efficiency. Enzymes are highly efficient catalysts, meaning that they can accelerate reactions by many orders of magnitude compared to the same reaction without an enzyme. This efficiency arises from the fact that enzymes lower the energy of activation required for the reaction to occur. Regulation. Enzyme activity is often regulated by factors such as temperature, pH, and the presence of cofactors or inhibitors. This regulation allows cells to control the rate of specific biochemical reactions and adapt to changes in their environment. Reusability. Enzymes are reusable catalysts, meaning that they can catalyze many rounds of the same reaction before they are degraded or denatured. This allows enzymes to be highly effective at catalyzing reactions even at low concentrations. Enzyme kinetics. Enzyme-catalyzed reactions follow Michaelis-Menten kinetics, which describe the relationship between the enzyme concentration, substrate concentration, and reaction rate. This kinetic behavior arises from the unique binding and catalytic properties of enzymes. Overall, enzymes are essential components of living systems, catalyzing the vast majority of biochemical reactions that occur in cells and enabling the complex processes of life. Types of enzymes. Oxidoreductases. These enzymes catalyze redox reactions, where electrons are transferred between molecules. They typically have two substrates, one that is oxidized and one that is reduced. Examples include alcohol dehydrogenase, which catalyzes the oxidation of alcohols, and cytochrome P450, which is involved in the metabolism of drugs and toxins in the liver. Transferases. These enzymes catalyze the transfer of a functional group, such as a phosphate group, from one molecule to another. Examples include protein kinases, which transfer phosphate groups to proteins, and DNA polymerases, which transfer nucleotide groups during DNA replication. Hydrolases. These enzymes catalyze the hydrolysis of a molecule, where a water molecule is used to break a bond. Examples include amylase, which breaks down starch into glucose molecules, and lactase, which breaks down lactose into glucose and galactose. Lyases. These enzymes catalyze the removal of a group from a molecule or the addition of a group to a molecule, without the involvement of water. Examples include carbonic anhydrase, which catalyzes the conversion of carbon dioxide to bicarbonate ion, and fumarase, which catalyzes the conversion of fumarate to malate. Isomerases. These enzymes catalyze the rearrangement of a molecule's structure, converting it from one isomer to another. Examples include glucose 6-phosphate isomerase, which catalyzes the conversion of glucose 6-phosphate to fructose 6-phosphate during glycolysis, and triose-phosphate isomerase, which catalyzes the interconversion of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Ligases. These enzymes catalyze the joining of two molecules using ATP. Examples include DNA ligase, which joins DNA fragments during DNA replication and repair, and RNA ligase, which joins RNA fragments during RNA processing. Kinases. These enzymes catalyze the transfer of a phosphate group from ATP to a molecule, typically a protein. Examples include protein kinase A, which phosphorylates proteins involved in cellular signaling, and glycogen synthase kinase, which regulates the synthesis of glycogen in the liver and muscles. Phosphatases. These enzymes catalyze the removal of a phosphate group from a molecule. Examples include alkaline phosphatase, which removes phosphate groups from a variety of substrates, and protein phosphatase 1, which dephosphorylates many proteins involved in cellular signaling. Proteases. These enzymes catalyze the hydrolysis of peptide bonds in proteins, breaking them down into smaller peptides or amino acids. Examples include trypsin, which cleaves peptides at specific amino acid residues, and caspases, which are involved in programmed cell death. Lipases. These enzymes catalyze the hydrolysis of lipids, breaking them down into fatty acids and glycerol. Examples include pancreatic lipase, which breaks down dietary fats in the small intestine, and hormone-sensitive lipase, which is involved in the breakdown of stored fats in adipose tissue. These are just a few examples of the many different types of enzymes that exist, each with a specific function and classification. Enzymes play a critical role in regulating and catalyzing specific biochemical reactions in cells and are essential for life.